Welcome to a new chapter of Glioblastoma, aka GBM, presented by the Glioblastoma Research Organization. This season, we're adopting a documentary-style format to offer you a never-before-seen look into the world of glioblastoma from around the globe. Join us as we venture into labs and companies worldwide, providing a comprehensive view of some of the cutting-edge efforts to combat GBM. Through intimate interviews and detailed on-site explorations, we'll offer an insider's perspective on the innovative strategies and real-time progress in the fight against glioblastoma. Thank you to our sponsors whose generous support has made this season possible. Please note that the sponsorship of this season does not constitute an endorsement of any sponsor's products or services. Remember, the information presented is for educational purposes only and it is not intended to treat, diagnose, or prevent any disease. Always consult your medical provider and healthcare team with any.
Hi, my name is Maria Vinci. Everybody knows me as Mara Vinci. I'm a cell biologist and a team leader of the Pediatric high-grade glioma group here at the Bambino Gesù Children's Hospital. In 2017, I joined here the Bambino Gesù to establish my group, and I trained before for many years in the field of pediatric neuro-oncology, and in particular focusing on pediatric high-grade glioma. So about the Bambino Gesù Children's Hospital is the largest European pediatric hospital, and it was founded in 1869 by the Salviati family, who donated then the hospital at the Holy See in 1924. The Bambino Gesù is located on a different site, with the oldest and historical site on the Gianicolo Hill, while here at the San Paolo we have the outpatient and the research labs that today we will feature also, and will tell you about our research on pediatric high-grade gliomas. The Bambino Gesù is a full pediatric hospital that focuses on different specialty from medical and surgical, oncomatology, neuroscience, transplantation, genetic and metabolic disease. But today we will focus our story on the pediatric high-grade glioma research that we conduct at the hospital. Within the neuro-oncology unit that is headed by the Dr. Angela Mastronuzzi, every year we see about 100-130 patients affected by tumors of the central nervous systems. These patients are in the vast majority about 5 years of age or below this. And in the past few years we see an increase in the number of also patients in the adolescent age and also young adults. In particular, regarding the patients affected by high-grade glioma, we see about 20 patients of these every year at the Bambino Gesù, and these patients are taken care, and the tumors are studied and characterized through next-generation sequencing, classical pathology, molecular profiling, and methylation profiling. And this allows really to provide the best care to our patients, alongside with the research activities that we conduct in the lab. It is really essential the collaboration that there is really between the clinical and the research activities, and that take place, for example, every week on tumor board meetings where clinicians, radiologists, neuro-oncologists, neurosurgeons, pathologists, and biologists discuss the cases and review the new cases as well as the follow-up cases. For the activities that we conduct in the lab, it is really essential this close collaboration, because differently from other research centers, one of the main advantages of conducting research here is really having access to patient material, which is really critical. The type of disease we are talking about, that is, although very aggressive and fatal, is still a rare disease. So my lab is, as I said, focusing our research activities on pediatric high-grade gliomas, and we have a number of few important questions we would like to answer, and we are currently answering with our ongoing project. One of the most important question we have on these tumors is how we can tackle tumor heterogeneity. Tumor heterogeneity is a particular difficult part of the biology of this disease that affects not only different tumor types within the family of the pediatric high-grade gliomas, so the inter-tumor heterogeneity, but as well as that, also inter-patient heterogeneity within the same tumor subgroups or intra-tumor heterogeneity. So how we are trying to address that? We are trying to tackle these problems combining and taking an integrating approach of a multimodal treatment and conducting research combining, for example, on ongoing research project, immunotherapeutic approach with targeted therapy. Another important question we have is about how can we stop and inhibit cell motility. These are very aggressive tumors that really diffuse within the central nervous system. And so we are trying to understand how these tumors really infiltrate the brain and what role has the cell-cell interaction into this tumor cell invasion and the tumor cell migration in the brain. Other important topic to address are how can we track the changes of these tumors over time. Unfortunately, it's difficult to intervene during the tumor progression, but we may take advantages of new approaches, for example, using a liquid biopsy, taking advantage of studying extracellular vesicles, or tumor and immune phenotyping to try to understand how the tumor changes over time, also under therapeutic pressure, and try to modulate this over time. And lastly, what we would like to try to address is to predict in a better way in vitro using the precious patient-derived models that we are able to establish and use within our lab, really to predict in a better way how to treat these tumors. We are, through collaborative studies, we are using microfluidic devices that can allow the pharmacokinetic profiling applications, and so providing more information about the dynamics change of treatment compared to classical static treatment that normally is performed in vitro. As I mentioned, one of the central pillars in our lab is the development and establishment of patient-derived cell models. So these have a unique complexity, but as well as the ability to retain features and molecular and phenotypic characteristics of the original tumor. So as you can imagine, the diagnostic part is always to be prioritized, but whatever material is left, we can use it in the lab and really make the most of it, establishing really primary patient-derived cell lines. One of the first things we can do with this is really to authenticate these, really using patient blood samples or tumor samples, really to define the authenticity of the cell line to the patient from which it was derived. And besides that, the most important part is the molecular characterization from DNA sequencing or methylation profiling that we can conduct here, or that we are really conducting through collaborative studies with other research centers that allow us really to demonstrate the reliability and the important role that these patient-derived models have, as they are so importantly resembling molecular features that are present in the patient tumor. There are different complexity on the patient-derived models. We can either use the cell lines in culture in vitro, or we can add additional complexity working with them in co-culture system and looking at how they interact with their other additional important element of the brain, like neuronal cells or endothelial cells. Additional complexity may be represented by using these to generate brain tumor organoids, so like really culturing in vitro little brain and trying to understand how these cells really interact in this in vitro microenvironment. And then additional level of complexity is, for example, taking advantage of organotypic brain slices, or then finally use these to implant orthotopically in the mouse brain and generate orthotopic mouse models of a specific molecular subtype or specific location. This is very important because it gives us really the possibility then to validate certain findings or test a particular treatment that otherwise we would not be able to test directly in the patient. A lot of effort in our lab currently is really on the application of spatial biology, and we are doing this using an advanced technology that is called imaging mass cytometry and single-cell proteomics. We have really set up the Hyperion lab here at the Bambino Gesù, where we can use the spatial biology approach to really providing us a new view of really the information and the heterogeneity and the tumor ecosystem that are within patient tissues. This is very important because it can really complement on a completely different way the more basic information that we can gain from bulk sequencing, for example, where we have just a general average expression, or even single cell sequencing data where we have expression at the single individual cells level, but we do not have in both cases the important spatial and the topographical information that instead with the spatial biology is retained in the tissue. This may be very important to really address a specific question in terms of trying to understand what is the composition of the tumor, where the cells are really located, and how they are interacting, identifying specific niches that may represent specific vulnerabilities that we can then attack in a more easy way by really adding this level of knowledge. This type of approach can be also very important also for our preclinical research. In the translational research and efficacy studies that we can conduct also in vivo, we can then really studying at a with a longitudinal approach and trying to understand, really taking the approach of the spatial biology and to try to understand in the mice how these preclinical tests that we perform are really affecting the tumor in situ. Another advantage of this specific application compared to other spatial biology techniques is that the same instrument can give us the possibility, for example, to perform single-cell proteomics on cell suspensions. So we may profile, for example, blood samples from patients or blood samples from mice and try to understand how that immune profiling can change over time, over treatment, and this is really giving us a new dimension to the knowledge that we had currently so far. Another important approach that we use to study especially our patient-derived models and the experiments that we perform is taking advantage of technologies like high-content image analysis. we have access to some of those platforms that really can give us really large information and giving the possibility to test really at a multi-scale a large number of compounds at the same time, or testing different cell lines all on the same microwave plate. And the advantage of using this type of platforms is that we can design specific assays, cell-based assay, so we can interrogate and perturbate our cells with the specific compounds or a library of compounds and see what kind of effects these produce. We can analyze by automation and high content image analysis, cell viability, or even the type of changes that in the cell morphology, and quantify this on a large scale. This is very important because then hits that comes from this type of analysis that can be further validated and then be taken forward for more advanced in vitro and then also preclinical study that can really taken forward also through in vivo testing. An interesting opportunity that this type of high content analysis can provide is, for example, going back to the point of how these cells interact. We can use this platform really to perform single cell tracking and really study the speed of how the cells move, or how many times they touch each other, or really which cell populations are really more closely related than others. So having this possibility really give us the opportunity to first of all establish this assay and then go and try to perturbate these interactions, since one of the opportunity in the treatment of this type of tumor is really cutting the cell-cell interaction to really make these tumors weaker. So in this case, for example, what we can see on this video is the co-culture of two individual clones that were isolated and expanded and then put in co-culture, derived from the same bulk GBM cell line. And here we can see over the video how the cells interact with each other. In the video you see two different colors, one that looks yellow and the other one is pink, and because the clones were optically barcoded. So we have used a specific technique to be able to track over time, to label over time, and to be able then to distinguish exactly the two different cell populations. And by doing so, then we can really add some layer of analysis using high content image analysis, because cells could be tracked over time while moving, and by doing so we can really quantify. So it's not just beautiful images, we can also quantify how many times cells interact, or how the cells move more quickly when they are individually cultured or when they are in co-culture. And this is very important because through this study we could demonstrate that when cells, heterogeneous populations that are present within the same tumor, are co-cultured, the interaction between these cells make the tumor stronger and more actively motile. So thinking about the future, one of the things I would like really to achieve is contributing to, for example, generating a single-cell spatial proteomics atlases of pediatric high-grade gliomas. We have in our hands the capability to master this new technology with the spatial proteomics and imaging mass cytometry that has been really included within the method of the year in 2024. I think this will be very not only interesting but useful for the scientific community. Besides this, from the treatment point of view, I think it will be very important really to move forward towards a more complex multimodal therapeutic approaches, and I'm really hoping and dreaming that some of the research projects we have currently ongoing in the lab will get closer and closer to the clinic and potentially one day be tested in our patients. I started working on pediatric high-grade glioma in 2012, and actually that was the year where some really pioneering discoveries have been made in this field, really that opened the way to really distinguish these tumors from the adult cases. There has been really a huge really advancement in the knowledge in the past few years. And we are really seeing with new clinical trials and innovative studies how the knowledge that we have gained in the past few years is really transforming this field. I really hope that in the next few years important collaborative studies will really make an important impact on the outcome of the patients affected by this aggressive disease. We know these are devastating disease and the cure is not just around the corner, but maybe with the research that is ongoing and additional research that will highlight new approaches and the new treatment in the next few years, we will be able really to improve the outcome, maybe to transform this disease in a more chronic disease instead of really such a devastating disease with a poor outcome for these patients. Unfortunately, what we are missing at the moment is really a view on how the tumor is changing over time. I think that by adding this level of information, if we are able in the next few years to really walk along the tumor changes and try to adjust the therapeutic approaches along the way, we may be able really to make an important impact in terms of maybe not curing but better treating these patients.